Hi, everybody. StreamYard tells us we're live, but as I always do, I'm gonna wait till YouTube tells me we're live because StreamYard has lied to me before. So I don't trust it. We're just gonna wait a second here. And as soon as I see me and hear me, then we're good. And I see me and hear me. So we're good. Hello, everybody. Welcome back to Creation Myths. Tonight, this is going to be so much fun. As you can see, Sal Cordova is here, and we are going to talk about phylogenetics. So, Sal, for many of you, I don't think Sal needs much of an introduction, but Sal, why don't you introduce yourself and your channel, and then we'll just get right into it.
Well, thank you, Dan. I run the Evidence and Reasons for the Christian Faith video channel, and Dan gave me permission to rebroadcast this on my channel. I've tried doing simulcast before, and it just blows up my computers and the system. So to the viewers that will be watching me via recording on my channel, greetings. And let me just put in a good plug for Dr. Dan. As many of you know, that I've studied biology like Emery Moyna in secular schools. He's a grad student, I was a grad student, and it's like, well, you know, people that are going to teach us of God's creation, for the Christians out there that believe in creationism, we have to learn from people like Dr. Dan, and that's kind of what I'm hoping to accomplish today, just to see to show everyone what it's like to study under a secular professor. And I will say this, and I've gotten flack for saying this, but I'll say it again. There was a poll between me and Emery, and we said between Kent Hovind, Dr. Dan, or PZ Myers, who do we like? We unanimously voted for Dr. Dan, hands down. So
very short. It's a very short list, though. It's not a lot. I mean, 1 in 3. I don't know if that's distinguishable from random chance.
Yeah,
but I'll take
yeah.
it. I appreciate it.
So I'm a molecular biophysics researcher, but most of my life I'd been an engineer in the aerospace and defense industry. And then in 2014, I ended up working for Dr. Sanford, mostly on proteins. So even though Dr. Dan has been actually very critical of my boss, that was mostly in a field that I was, I'm not really a genetics guy. And I am a protein guy. So that is kind of where it goes. And, you know, there are areas that Dr. Dan and I will have to agree to disagree. But today, he was very kind to come to my channel, and I realized when we were interacting, I said he could actually teach me a lot. So that's kind of what I was hoping to accomplish today. I know some of my creationist colleagues don't exactly like my friendly terms, and I just like...
They don't like me. It's fine.
Yeah, yeah. So now they don't like me either. So that's kind of the situation.
Yeah, so it goes.
Yeah.
So, here, why don't you put up those slides? This is from like a while. This is from like a few months ago now that we like very briefly interacted. Yes. And then we were like, let's put a pin in that and talk about it later. And now it's like several months later. And later is finally now.
Yes. So my goal was in having Dr. Dan, it's like, well, we want to, you know, I want to be able to say, okay, this is how I describe evolutionary theory. And if it's not accurate, I invited Dr. Dan to say, no, that's not accurate. So at least when I teach creationists what evolutionary theory says, I want to be accurate there. And then also, you know, if I'm missing some things on basic biology, that's important. So Dr. Dan actually helped me, like introduced me to OpenStax, and I'm now giving lectures from OpenStax. In chemistry, I haven't gotten to the biology, so I'm indebted to him for that. So I'm going to show the first slide that he flagged, and that kind of began the conversation. And it's this one here. It follows Haeckel's.
There we go.
From origin of life to humans. And this, I actually just liked it because of the graph. And he said, well, there's some problems with it. And I said, yeah, I know there's some creatures there, like I know the font's kind of not too clear, like blastula and gastrus something over here in the in this region here. And we won't really have much to say about that, but he was amused. He said, "Yeah, Sal, don't use that. Don't teach
Right.
creationist evolution with that." So that was the
Right.
beginning.
Because the problem here is that a lot of the things on this figure are alive today. Now, I can't read specifically what everything is, so I think some of this stuff is extinct. Like I think if you look kind of in the, you can see the numbers. So I think like kind of in the middle there, like maybe 15, maybe number 14. I think somewhere in there, maybe some of that stuff is extinct. And then I'm not sure what's going on down in the middle with like 21, 22, 20. I'm not sure which of those are supposed to represent like kind of the ancestral common ancestors for like mammalia and which of them are supposed to represent things that are alive today. But what you do see here is a lot of progression between things that are alive today. And one of the first rules with phylogenetics is that nothing that is alive today is descended from anything else that is alive today. So if we're going to have like a chain of evolutionary history, nothing in that chain except for the thing that's alive at the end of it can be alive today, right? And you see a lot of that here where I think there's marsupials in here, and then it goes into placental mammals, and it kind of goes through different morphologies, and you end up at like kind of going through monkeys and then apes and then into like derived apes on two legs. And it's like, well, that's not how it works, right? It would have been, it's not, you can't use living monkeys to represent a common ancestor of living humans, right? It's just that's the first thing. So that's why I recommend against a figure like this, because it shows it as a linear progression where there are things that are alive today at different stages of that linear progression, which is not how phylogenetics works.
Now, this actually raises an interesting question because you showed that radiation diagram where you said it's like a tree, but it's just branching and branching. And I did want to find a diagram where we could go from, like, say, a primitive creature all the way to humans. And so it basically, you would actually just kind of, you'd say that the rest of the branches are there, but you're just going to focus on one. And I don't know if they have something like that, because I used to see these progressions where you go from
I actually, I'm going to see if I can find, I'm going to dig up some stuff I use in class, because we actually do this in class every spring. In my class, we start with the unit on biodiversity, and it's all about cladistics and phylogenetics. So it looks at, so basically the way we teach it is we start with prokaryotes and we kind of go and we end with human evolution. And the things that we emphasize at each thing, it's not what was that organism at each node, at each branch point on the tree. It's not like connect the dots through what do the organisms look like. It's all about traits. So you start with, you know, your ancestral heterotrophic prokaryote, anaerobic, and then you kind of work your way forward in time, layering on more and more derived traits as you go, right? So a nucleus and multicellularity and blah, 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 all the way down to like a really big frontal lobe all the way at the end of the line. So I'm actually going to see if I can find this real fast, because I should be able to find this real fast. And that will, I think, be potentially useful for us. So give me just 1 second. Well, please, if you want to show another thing.
Okay, sure, great.
Please.
That's great.
And while I'm just kind of looking around for this phylogeny that we use, or phylogenies.
So I'm going to stop and I'll just talk about my background here for just to give context. So I actually do a little bit of, I had to learn this as part of my bioinformatics class when I was at the FAES graduate school at the NIH. And by the way, Cy Gart is speaking. He'll be on Debate, Modern Day Debate. He was a professor at my school. So I think that'd be interesting. Someone asked, is Sal a YEC or just a creationist? I am a young Earth, young cosmos creationist. I used to be what we would call a theistic evolutionist, and then an old Earth creationist for most of my life, and then a young life creationist. And you know, there are all these varieties. I had a big show on all the varieties of thought, kind of what you'd say on our side of the aisle. And so I was a young life creationist, kind of like agnostic on the age of the universe. After graduate school at Johns Hopkins in applied physics, I declared myself as a young cosmos creationist, which means I also believe the universe is young. So, and then I ended up, so this is really strange that most of my professional life, I was in the aerospace, defense industry, engineering. So I'm relatively new to biology. And when I started working for Dr. Sanford, he sent me off to the FAAS school at the NIH. And I know I'm going to get some flack for this. He didn't send me to Dinosaur Adventureland. And I studied under an evolutionary computational biologist there who was actually on the staff of Eugene Koonin. They have an evolutionary genomics group there, about 30 evolutionary biologists in one group, and I think that's pretty large. And Koonin's a recognized name, and so my professor co-authored articles with him, and that was a course on bioinformatics, and that's where I began to learn kind of the phylogenetics. He was showing through the phylogenetic software and bioinformatics. Now, I've interacted with Joe Felsenstein. I've published in population genetics, but that has been mostly in connection with Fisher's fundamental theorem of natural selection and the idea of fitness. So there are all these bodies of computational work, and I actually know, like, they're terms that I was seeing, and I'm like, I think I need to know that, because to be at least competent to be able to describe. So like when I showed the human evolution thing that Dr. Dan was pointing out, he said, like, hey, I don't show that. It's like, yeah, I need to learn stuff like that, because I actually, I never had a formal evolution class except those 3 weeks studying phylogenies. And then I actually know stuff more from the mathematical side. This is Joe Felsenstein's Inferring Phylogenies, and it's a miserable read.
It's like the math of phylogenetics is dense. It's like, it's really hardcore, especially if you get into like the really computationally demanding techniques. So you get into like the Bayesian techniques, and then if you really, I mean, you could just set up the computer program and run it without like kind of understanding the underlying math, or you could get into the underlying math, and then that gets really intense really fast. Which is not to say it's not interesting or enjoyable. It's just computational. Like, it takes work to understand the underlying math of what's allowing you to do this.
Yeah, no slight on Dr. Felsenstein. I said it was miserable because it was so mathematically challenging. It was, one could almost look at it and say, what does this have to do with evolution? It's doing a lot of probability computations and things, very, very detailed. You know, I said, okay, I can appreciate it as a creationist for the math. And that was, you know, I had to try to find motivation. You know, I mean, honestly, it's challenging when one doesn't accept the theory and you're still studying it. It's like you have to find motivation more than just trying to abuse the theory. You know, just try to find something redeeming or something you can appreciate. And so it was the math. So thank you for this diagram. Oh my goodness.
Yeah, so this is actually, for everyone watching, this is actually what I use in class. So we do this unit on biodiversity, and it's in a phylogenetics and cladistics concept. So it's all about monophyly and nested hierarchies. And the way it's set up is the derived traits that get you from your ancestral to your derived states, right? So what we do is we look at all these living groups, and we do this from very broad to very specific, right? So you kind of go through these three that I have right here. This is the eukaryote tree. This one changes every year because we're constantly rearranging what the supergroups are within domain eukarya. So I'm going to just put this one aside because it's a giant mess. This one is a good place to start because this is animals, and we generally know like what these different group, like people watching will probably generally know what these groups of animals are. And if you're not sure, easy way to remember is there's a SpongeBob character for every phylum on this tree, basically. So if you've ever watched SpongeBob SquarePants, you can just go down the line and you get a character for every branch. So the thing here is, what you'll notice is we have two sets of things, the way we have the students kind of practice this stuff. We have the numbered groups, so those are going to be your taxa, and they can be like the broad clades like Metazoa, which is synonymous with animals, or they could be specific down. All these groups are phyla right here, not quinoflagellates. They're a different thing. They're the outgroup. So then you have, your porphyry, your sponges, your cnidarians. I'm leaving out a bunch. Like there's other stuff in, like there's the ctenophores in there and the placozoa. We don't actually know the arrangement of like porifera and those other groups really, like up here. We don't. So this is a simplified version. There's way more phyla than this. There's like, I don't know, if other people were here that are more kind of familiar with animal phylogenetics, they could tell everybody how many phyla there are. There's like 30 something, maybe 40 something. I don't know off the top of my head. We just hit kind of the big prominent ones. So the important thing is the letters. Is the letters for each node, the letters are where I want the students to know the derived traits that allow you to group each clade in the nested hierarchy. And for everyone listening, the word clade just means monophyletic group. So that's a group that includes a common ancestor and all the descendants from that group. So like animals, or on this tree, it's metazoa, right? It's metazoa. That is a clade. And then right here at A, this node right here, node equals branch point. So this branch point right here, that represents the most recent common ancestor of all animals. And we can say, okay, what derived traits are there? Like this is what I want my students to be able to do. They actually get the blank version. I say, okay, you need to be able to fill this in.
Dr. Dan, if I could interrupt you.
Yeah, go ahead.
This is mostly for questions.
Yeah, go ahead, please.
That actually was one of the reasons I wanted to confer. I actually had problems understanding the concept of derived traits.
Bingo.
And then also, could I show at least one, before we flip back to this, could I show?
Yeah, here, put your screen back up.
I just wanted to show the group. I actually built a, so I'll share my screen, and this is probably the last time you'll see this, but I actually had to do this. I actually built this one partly in connection to my topoisomerase work on proteins. Now, what's really funny, everyone is so specialized. My colleague Joe DeWeese had worked for like 15 years in topoisomerase and the molecular biology. I was the one who showed him how to use this and build the trees, because we weren't really that interested in evolutionary progressions. He focuses on human topoisomerase because that's the target of cancer therapies, but we happen to actually just need other organisms to do comparisons, because we were doing some things that were related to what we were trying to publish. So it's like, oh, it's really good, Sal. You figured out how to use Mega. And so maybe later we could talk about rooted and unrooted. And like, I didn't know how to.
We can 100% talk rooted and unrooted. Yeah. So let me.
So let me just.
Absolutely.
I'm just going to stop sharing, and we'll. So I just wanted to say, okay, so this is the extent of what I've been able to do. It's like I throw in protein sequences, and they're easier than genes, and put it through Joe Felsenstein's algorithms, and it spits this out. I'm like, okay, I knew enough how to do that. That's it.
And so this is a rooted tree, right? So this is a rooted tree, meaning we have evolutionary direction, and we're using the E. coli right here as on the bottom there. That's your outgroup. So you're saying that's your kind of basis of comparison, right? And then you're saying, okay, everyone else is more closely related to each other than any of them are to E. coli. And then we're grouping them. I don't know if this is, I'm guessing this is a maximum likelihood tree, is my guess, because that's kind of a baseline way of doing it. Do you know what the underlying?
I actually don't remember. I kind of like the UPGMA just because it's such a cool name, but even though no one uses it, I don't see any publications that ever use UPGMA. So I was just being kind of a rebel here.
I'm not sure what that would be. I'm not familiar with that one off the top of my head. So,
but I mean, we have all the settings. But the thing is see this, and I want to be just transparent here. It's like, okay, I need to know how to read these diagrams a little better, because the professor said, he would say, okay, we were flipping through pages, and he said, this is mid-rooted, this was, this is rooted, this is unrooted. And of course, this relates to arguments that are happening over Jeanson's work. And I said, hey, time out. We need to get all this straightened out. Yeah, at least I do. And so in the thing is so much of bioinformatics, when we're doing classifications of proteins, it's based on phylogenetics. And so we kind of got it down from the most of the bioinformatics was talking about the comparison algorithms and homologies and the computational methods and all the alignment algorithms. So we got it from the bottom up, and then we just had little piecemeals of phylogeny here. So I actually was surprised when you said this is a rooted tree. So I learned something because I didn't set rooting on my in my switches, and I said, okay, I don't know then what I'm doing. So that's why I said I need to talk to Dr. Dan. So anyway, that's about all. That's the extent.
So first thing, I want to actually address a really interesting conversation going on in the chat before I switch over to rooted versus unrooted, and it's whether things are in the wrong place on this tree. And I think that's the wrong way to look at it, because each tree, you make trees based on specific genes or proteins or regions of the genome or whatever, and within a given tree, you generate the best tree based on whatever technique you're using. Andrew says that this tree is based on genetic distance, so it's similar to neighbor joining. Neighbor joining is like kind of a mediocre technique. There are better techniques now, but like it's fine. It's an intuitive way of doing it where you just kind of pair up the most similar sequences and do that iteratively. Really pretty straightforward technique. And the thing is, there's nothing necessarily wrong about any given tree, because each tree is essentially a hypothesis where you're saying, okay, based on this specific sequence, here's what we think the relationship is. And then we can compare other sequences and see if they are consistent with that hypothesized relationship or if they are inconsistent with that hypothesized relationship. And by looking at a bunch of different gene or protein sequences or whatever, you can build a consensus and say, okay, 95, 90, whatever percent of phylogenies involving these 5 species generate this pattern. That's what we think the pattern of descent is for this particular group, right? So it's not that anything is out of place. It's that different relationships on the tree may be consistent with other findings using other parts of the genome, or they may be inconsistent with those things. I just want to point that out to everyone, that you're going to get, as someone said, you're going to get differences between different genes. You're going to get differences between gene trees and species trees. You're going to get huge differences when you do protein trees, because amino acid sequences can vary very widely and end up with approximately the same function. So you have a lot of variation. So protein trees end up kind of weird. And you can see an example on this tree where the plant is more closely related to all the animals. You can see in the middle here, it's right above the second red arrow from the bottom. You see you've got Arabidopsis in there. That's weird because plants are phylogenetically more distant from animals than fungi. And so yeast in between those bottom, actually the second red arrow from the bottom, that's yeast right there, Saccharomyces cerevisiae. You'd expect that to be closer to animals. It's weird that it's not compared to the plants, but that happens sometimes just because, especially with protein trees, you get so much variation in them.
And it's interesting you mentioned that. My professor, he actually, Rogozin is his name, Igor Rogozin, and he published, he with Kunin published something that actually said something interesting about Arabidopsis in humans. And he said, well, if they did an intron tree, and they said, well, we got some strange results. The Arabidopsis is closer to humans than other animals like C. elegans and
weird, yeah.
a Drosophila.
That happens, yeah.
So I just wanted to ask, when you were saying it looked out of order, does that mean it's like, to me, is saying that depending on the way you approach this, like you said, between proteins versus species, and then even the algorithm that you use to build the tree, it seems to me you're saying that you would actually generate different clades.
Yeah. Oh, 100%.
That's okay. That's another alternative way of saying it.
And in fact, within single techniques, you can do the same analysis multiple times and get different trees because they're not deterministic. So a neighbor joining tree is deterministic. You do neighbor joining, you set up the parameters with the same data set. You do it 100 times, you're going to get the same tree 100 times. You do a Bayesian analysis on that same data set 100 times, you're going to get 100 different trees. They're going to be similar to each other, but you're not going to get the same answer every time because many of these techniques, they're not deterministic, they're probabilistic. So you don't get the same answer each time, but the point is to do it iteratively and do it for lots of different regions of the genome, so you converge on a consensus. You don't want to put all your eggs in one basket in terms of what part of the genome do you think is most informative. You do it really widely. You get a bunch of different but similar answers, and you kind of build a consensus based on that.
And so see, and I kept hearing the term bootstrapping, and I actually could turn on the bootstrapping option. And my understanding is one of the challenges is that computationally, it'd be nice if you could just sample every possible tree, but that's not combinatorially possible.
Right.
Then you take samples randomly, and then you slowly build your consensus.
Do you get the same tree? How often do you get the same specific node? Right. That's exactly how bootstrapping works. Simplifying the math, but it's basically instead of taking your whole data set, you're sampling from it piecemeal and saying, okay, with a subset, do I get the same results? And that's a test of kind of robustness of your conclusion. Is it something that's robust across my data set, or is it something that's biased by which fraction of the data set I'm using? And if it's biased by which part, if, for one node, you only get that node 50 or 60 or 40% of the time when you're bootstrapping, then that's not such a reliable conclusion. If you get the same thing 90, 95, 99% of the time, that's a more reliable conclusion.
See, this is great, and I don't want to hold up the discussion here because you had such a good thing on mammals. But like when I was flipping through Mega and getting all these trees, I said, okay, there's one that gives numbers and then one that doesn't.
That's the bootstrap one, yeah.
Yeah. So the UPGMA, see, you clarified something. They're deterministic algorithms. So you said neighbor joining UPGMA or deterministic.
I don't want to say UPGMA is necessarily deterministic, but Andrew said it's similar to neighbor joining, and I know neighbor joining is deterministic. I don't want to get out over my skis on that one, but then the other ones are not, right? So so here, let me pull up. If we can just change gears and go back.
I'll shut this one down here.
Here, so let me pull up this one. This is real. So what I just want to show here is just the rooted versus unrooted trees, because this is something that's confusing, right? So when you root a tree, the point of what you're doing is you're giving it an evolutionary direction. You're making a judgment call and saying, this is the ancestral state, this is the derived state. And based on that, we can say, okay, here's where we're going to start our tree, and then we're going to evolve from that point. So this tree right here, which is a very simple tree, it has 4 taxa, A, B, C, D, and the branches are color-coded to make it easy to understand once we root it. And then we have one node right here connecting A and B, and we have a second node over here connecting C and D. Now, even though we don't know what traits that we use to make this tree, we don't know which ones are ancestral or derived because it's an unrooted tree. So we're not saying this state occurred earlier, then it changed into this state later on. We're not saying anything about that. We're just comparing the similarity, basically, between the 4 of these things. And what we can learn from this unrooted tree is that A and B are more closely related to each other than either of them are C and D. And C and D are more closely related to each other than either of them are to A and B. That's what we get out of the unrooted tree, just in terms of gross, how different are you from each other? That's the information we can get here. But we can root this. Oh, go ahead.
I'm sorry. There are terms I probably need to review, and I'll just throw out. I really never was taught ancestral versus derived traits.
All right, let's see if I can find this in here. Here we go. Here we go. I got a slide for that. Don't worry.
Okay.
Got you covered.
Great.
Okay. So an ancestral trait, I threw these slides together like half an hour. This is a mix of stuff I use in class and a mix of stuff I've used on this channel. So it's it's a weird mix. But so this is just from class. And so an ancestral trait is also called a plesiomorphy. All it means is it's something, whatever group you're looking at, species, phylum, doesn't matter what it is. It's just whatever you inherit from a common ancestor outside of your group, that is an ancestral trait for that group. So
if you could give some examples, and then could we you used words like group and phyla? Are phyla also clades?
Yes. So I can get to that next. So let's do ancestral derived. It's no. This is great. This is great. We want to make sure we're exact. Start with the foundation and go up from there, right? So, first thing, these terms, ancestral and derived, are relative terms, right? You can't point to a trait and say that's derived in an absolute sense. It's always relative to whatever organisms you're considering, right? So, like, you could look at humans, or here's the example on the screen, right? Birds. So you look at the bone structure of the wings for birds, right? They have a humerus, they got an ulna and a radius. They're missing most of their fingers, but they have like carpals and metacarpals in there, but they're missing most of the phalanges, right? So you can look at that bone structure in the birds, especially the humerus, the ulna, and the radius, and you can say, okay, that's ancestral because birds are tetrapods. You go back to the ancestral tetrapods, mammals are tetrapods, amphibians are tetrapods. We all have that same bone structure. You could even go to some lobe-finned fish, right? Like coelacanth has a humerus and ulna and radius. So that bone structure existed earlier in evolutionary history than the clade birds, right? So we can say that's an ancestral trait for the group that we call birds. Does that make sense?
I'm just trying to process it. So it doesn't necessarily it's an ancestral trait. It doesn't necessarily define the clade.
It specifically does not define the clade because it existed earlier than the clade itself. So I'll give you another example.
Really helpful. So it's seeing that we have these plecomorphies, and I just wanted to say something that was funny because if Erica's here, it was really funny when I was on her show. I said, "Have you ever heard of Hennig, Willie Hennig?" She said, "No." And I'm like, I think that these morphe terms like plecomorphy, synapomorphy, that was from actually Hennig, and it's kind of it's kind of a funny relationship with the evolutionary community. They're kind of in their own, but some of the language. So that would be interesting. Anyway, that's a side note. So this was very, very helpful.
Cool. So
because the wiki entry didn't help me. You've helped me more than the Wikipedia entry. This is way better. Well, you ought to be a Wikipedia editor for that section.
So, you practice explaining this enough times, you kind of get used to it. So because it's not intuitive, these terms are not intuitive, right? So I'll give you another example: a nucleus, right? A nucleus is an ancestral trait for humans, right? Because you think about like all eukaryotes like have nuclei, right? So humans, like our cells, have nuclei, but that's not a trait that defines humans. It's not new in Homo sapiens. Right, it's a plesiomorphy. It existed way before we did. Right.
So this is, hey, I can feel really sophisticated now. I can use these terms.
All right.
So the nucleus is a plesiomorphy of humans.
But let's go to derived traits now. So derived trait is something that's new for your group, and you can just ignore characters or character states. That has to do with something I talked about earlier that I showed this slide in class. Everyone can just ignore that line. Just forget that. So derived traits or apomorphies are, if you're looking at, again, a group, you're looking at a specific group because these are relative terms. So a derived trait is something that is new in a particular group. So like
opposable thumbs, would that be a derived trait?
Opposable for like some primates.
That's it. Okay.
Right. So you've got your most ancestral primates don't have opposable thumbs. They have like 5 fingers kind of in a row that can grasp, but they don't have opposable thumbs. And then I don't know at what level you get opposable thumbs, but at some point it might be simians. Somebody in the chat, I'm sure there's people here that know this better
where are you when we need you?
But there's someone at some point, it might be simians. I don't know. I'm sorry, I'm not a primate person, but I mean, I'm a primate, but I'm not a primate evolution specific person. But at some point you get, they have opposable thumbs, and that's a derived trait at whatever level that happens. The example on the slide, wings in birds, right? So you have the limbs in birds is an ancestral trait. Go back to the common ancestor of birds, the common ancestor that birds share with amphibians, further back, that thing has that limb. But that limb existing as a wing, as a flight structure, that is a new trait that only exists in the clade of birds. So that's a derived trait for birds.
So apomorphies define a clade. Is that
we're going to get there? So there's one more word. There's one more word. So because derived trait apomorphy is just a very, it's a very broad term, right? And again, it's all relative. So you could say in humans, right? So let's compare humans to bacteria, right? So for humans, a nucleus is a derived trait relative to bacteria. It's based on the comparison.
Okay. Okay. This is, I have to process this. This is where I was getting hung up on the Wikipedia article and every place else that I tried to learn. So you're saying it is so a nucleus in a human is a pleomorphy, but then it can also be said it can also be argued to be a derived trait relative to bacteria.
It depends on what you're comparing it to. All of these terms are when you're talking about two things and comparing them. It's not an absolute, there's no trait that you could say in an absolute sense is ancestral or derived. It's based on whatever things you're comparing at the time. They're all relative.
This is so helpful because that would mean, depending on what literature I'm reading, I could see it characterized a pleomorphy being described as an apomorphy. And it's not.
Depending on the context, then that'll be right. It totally context dependent. So then there's one more term that is actually really useful for us, and it's a synapomorphy. I'll just add that syn to the front of that word, synapomorphy. Now that is a derived trait that defines a clade, right? So then you can say, like, for example, let's look at angiosperms, right? Flowering plants. The presence of flowers and fruit, those are both synapomorphies for flowering plants. They are derived traits, so they're new in flowering plants relative to like the common ancestor they share with their closest non-angiosperm relative. And we can use the presence or absence of those traits to define that clade, right? If you have flowers, if you make fruit, then you're an angiosperm.
And so flowering is a the flowering trait is a synapomorphy. Is that correct?
Yes. So you would say for angiosperms, specific, like the clade of flowering plants, right? You would say the presence of, I mean, the name of the clade is flowering plants, right? Like it's kind of, it kind of tells you what the gist is there. So then like the presence of flowers, which itself has like a bunch of components to it. It's like, it's leaves modified into petals, and it's like various reproductive structures. Like there's very specific components of that. The presence of those morphological traits is, and I apologize to plant people, I'm going to oversimplify, but the presence of those specific traits is sufficient to say this thing is an angiosperm, right? So that makes it a synapomorphy for the clade that is angiosperms.
I'm going to have to process this. So that makes it a synapomorphy. It's basically an apomorphy that defines the clade.
That defines a clade, yeah.
Okay. All right. It's a trait. Well, it's a trait that defines the clade. Is that fair?
A derived trait that defines the clade.
It has to be derived.
And the specific way I explain it is It's a derived trait relative to the common ancestor a clade shares with its sister. So the sister group are the closest living relatives in evolutionary terms, right? So like in terms of like primates, humans and the genus Pan, which are chimps and bonobos, like chimps and bonobos are the sister group to humans, right? So the closest living relative. So we share a common ancestor with each other that's more recent than the common ancestor we share with anybody else. And you can define a synapomorphy based on that common ancestor and say it's present in the clade, but it's absent from that common ancestor you share with the sister group.
Okay.
Which I know there was a lot of words in there, and I apologize, both to you and everybody listening that's like,
now, if I could just throw this out, because most of my work is in the molecular realm, and as I was generating these phylogenies, if we're doing, and that was actually an interesting thing with this inferring phylogenies, and I got a backstory here. Joe Felsenstein actually had some confrontations with the Hennig school, which had all these terms of synapomorphy, and he was saying there's something with what did he call it? Molecular cladistics had some issues with the morphological cladistics. So
Yeah.
we use synapomorphies in the molecular world. Do we use that
Right.
at all?
Yeah, it's the same, right? So let's keep molecular to one side for a minute because it's way easier to kind of process when you're talking about morphological traits. And then we can kind of, once we kind of have that framework, we can flip it over and it applies to molecular, but it's obviously different when you're dealing with molecular traits. So I want to go back to something that you said a few minutes ago about I think, what was the phrase you used? Taxonomic cladistics or I forget the word you used. Systematic cladistics. Something. It was something with cladistics in it, and I forget the word you used. But I have a slide for that.
I probably just made it up actually. So there was an interesting. I used molecular either taxonomies or molecular phylogenies. Yeah. So there was an interesting passage here in Felsenstein's book where he's talking with the traditional morphological, and they're trying to build the morphological trees. And he said, well, this was way back in the 60s. They were starting to go to the molecular because we actually didn't have molecular phylogenies.
Yeah, we only started sequencing stuff in the I think we got RNA maybe in the 60s, but we didn't get DNA until the 70s. And the first DNA genome was a teensy little virus called PhyX174. Which is only about 5,500 nucleotides, and it's 5,500 bases, not base pairs, because it's weird. It's a single-stranded DNA bacteriophage.
Oh yeah, so he had an interesting chapter on that, and so I may have not used the terms. So this whole thing of phylogeny was long before the molecular side came,
right?
And so.
And it messes, we change everything once we have the molecular stuff. Like the trees I showed you earlier that I use in class with all different groups of animals, those are all molecular phylogenies. We describe them based on morphological traits, but they're no longer defined based on the morphological traits. It just is the case that there are some morphological traits that correlate with each group that are basically useful indicators of a clade, but we no longer define the clade based on those morphological traits.
Oh, if I could just read the paragraph that it's just so this is from Joe Felsenstein's book, Inferring Phylogenies. And this is where it's probably good that we're having this conversation because you'll hear one group argue one thing and another group argue another. And he said, advocates of the hypothetico-deductive and logical parsimony frameworks are united in one important respect. They reject statistical inference as the correct model for inferring phylogenies. And I'm like, oh my goodness, so I was saying that this is, by the way, page 143 of Felsenstein's book. And I did, it's like, gee, I did not even know this. And so I'm just like, okay, so now I'm realizing now that we have the people that study the morphologies and they're building phylogenies. And then we have it seems like we have the statistical guys that have a different approach. And I'm just throwing that out there. And so this was actually very, okay. That's where I'm coming from. And I'm getting confused because of this.
Gotcha.
I'm getting confused. So, that's basically the thing.
So let's take a step back though, because I want to go back to, you mentioned earlier, confusion about basically relating the taxonomy to the phylogeny. And this is like one of the sticking points that I always got to work on every year, right? Because this is again, not intuitive. So let's start with this, and it would help if I shared my screen maybe. So let's start with this right here, right? So we've got our Linnaean classification scheme. Everyone that studied biology learns this in high school, domain, kingdom, phylum, class, order, family, genus, species, yada, yada, yada. And then we talk about taxa as just a catch-all term for a group at any level or in between the levels. What we want to do when we're assigning organisms to any level in here and anything in between is we want the nested hierarchy of the taxonomy, how we name and group things. We want that to match the evolutionary history. So what we end up doing is taking our box in a box system, right? That's essentially what our taxonomic system is, right? Domain, right? And then you've got kingdoms within your domain. Within each kingdom, you blow that up, you've got phyla in there. It's a box and a box and a box and a box system where you start very specific at species or even subspecies or strain or whatever, and you go all the way up until you end up in like a domain, right? So Sal, are we okay with that?
Yeah, yeah.
So what we want to do is we want to take this system, this nested hierarchical system of classification, and we want to superimpose it onto a phylogeny so that you have a nested hierarchy based on monophyletic groups, which is to say a common ancestor like this node right here and all of its descendants, right? So that's a monophyletic group. And you can say, okay, each terminal branch, that's a species right here. And you can group this clade as a genus. This one over here is on its own. These 4 right here, we'll put them in a different genus. And then you could group these 2 genera together into a single family descended from this common ancestor. You could put these 2 families together into an order descended from this common ancestor, and so on all the way back. So the idea is you have more and more inclusive groups as you get bigger and bigger in your hierarchy. And as you do that, you're including common ancestors in the more and more distant past. And the way you separate the clades at each level is by looking at derived traits that allow you to distinguish them from the other branches. How does that feel?
Could we use the term synapomorphy then?
Yeah.
Define?
Oh, absolutely.
Okay.
Yes. So you could say like, okay, this genus right here in green, actually we'll use this one as an example. So this family right here, this family has a synapomorphy, right? 1 or more synapomorphies that you say are unique to this family. They are present in this common ancestor right here, but they are absent from the common ancestor right here with the sister group, this other family. So you could say that synapomorphy that all of these organisms share, plus their common ancestor, we think shared it, that we can use that trait as a derived trait that is specific to this group that we can say allows us to classify this clade as a distinct group from the other one, right? So at each level, you want to use synapomorphies to distinguish your different groups.
Oh, now I know why I brought up the thing with molecular stuff, because now it's becoming clear why there's been some problem with the molecular phylogenies. Is if we're only doing this for single protein, and a lot of times the algorithms work better for a single protein, it's not clear you even have a synapomorphy. Like the COX-1 protein is identical between chimps and humans, so you would actually not be able to. You will not be able to identify synapomorphies that distinguish the groups. Now I remember why that came up, because I can't always see it, and then also because of the funny things like what you said with the proteins. There are characteristics we share, like because I studied the topoisomerase with chickens that are closer than we have with other primates, and it's like, well, yeah, that can happen because I know enough of the. One position can mean a lot, and you'll actually see that, and that's actually an active discussion now in the topoisomerase community. How are we going to? And I see that it's like, okay, it's more ambiguous when you have isolated proteins. You can't really see the synapomorphies as clearly as if you have, like, say, morphology.
You're exactly right. It depends on the rate of evolution for specific things. So certain proteins, genes, whatever, are very highly constrained and very highly conserved over long periods of time. And a lot of that has to do with like stuff that everything does. So like all organisms with double-stranded DNA, or at least all eukaryotes, prokaryotes have topoisomerases too. So everything with double-stranded DNA uses topoisomerases, right? So like you look at topoisomerases, they're very ancient. You can tell mammalian from prokaryotic or bacterial topoisomerases, but if you look at human and chimp and gorilla topoisomerases, those are so tightly constrained, right? You have purifying selection operating on things like those, or like ribosomes, some other examples would be like some very, very tightly constrained like metabolic pathways where I'm thinking of stuff, not cytochrome C oxidase, but some of the other electron transport chain stuff is like really tightly conserved. So you can tell differences across like maybe phyla or kingdom level, but you're not going to tell differences across like genus level, that kind of thing. But there are other proteins that they're less constrained. So like hemoglobin, for example, you can tell species, right? You can tell human from chimp from gorilla hemoglobin, and you can tell mammalian from avian from non-avian reptile hemoglobin, right? So you can tell kind of different, because those are a little less constrained.
Oh, speaking of which, just for example, histones. Oh my goodness, 100% identity across.
Histones are very right, because histones are basically derived. So histones appeared basically for eukaryotes, right? So it's a derived, and I know one of your things is the prokaryote eukaryote transition, right? So histones, they are derived for eukaryotes. Prokaryotes
would they be
have
a synapomorphy for the eukaryotes?
So that would be a synapomorphy for eukaryotes, right? The presence of histones, right? Yeah.
Now, the thing, I mean, because that was what I was studying at the NIH was the histones, and they said it's so deeply, everyone is kind of like a known thing, and so you can't actually identify species. Like how, any animal has identical histones with a human. So you're not going to be
close enough that you can't get an informative phylogeny out of it. Right.
So,
but then there are other things that are not like that.
So and this is starting to clarify, yeah.
So when you're doing molecular phylogenetics, when you're using, instead of using physical traits, instead of using, the presence of a humerus, right? Or do you, how many digits do you have, right? Instead of using morphological traits like that, if you're using molecular traits, now it's a different ballgame, because the molecular traits change at different rates. And so looking at different molecular traits, you're going to be able to resolve your differences at different levels. So like if you want to tell domains apart, ribosomal DNA is a great candidate for that, because you can tell eukaryotic from archaean from bacterial ribosomes apart really readily. If you want to tell primates apart, ribosomes are useless for that, right? Because they're all too similar and you don't have enough differences to resolve your phylogenetic signal.
So would that be the 16S or the 5S?
Yeah, exactly. Right. It would be the 16 or whatever the other one is. Yeah, it's the subunits for the so the DNA that encodes the ribosomal RNA. Yeah.
Yeah. Oh, now I remember, because that was one that Carl Woese.
So Woese used that to establish the 3 domains. Yeah.
This is, I can't tell you how informative this is. See, now I'm beginning to understand why there were.
But then you can't use that to tell something, but to tell, like, if you want to tell, what's the pattern of evolution between orangutans, gorillas, chimps, and humans, ribosomal DNA is useless for that, because they're all too similar. You have too few differences. Okay. So use something that evolves faster. So you could use, what you would use for something that's recent is you would use some non-coding DNA that we think is going to accumulate mutations without purifying selection. So then you could actually see the pattern really nicely.
Yeah, see, this explains a lot, because a lot of evolutionary papers that I've seen, again, they're molecular. They really love
all molecular now, yeah.
Yeah, and it's like, okay, this is where I could see that there's a little bit of, it causes a little bit of problems, because sometimes they're only studying one protein to try to make a phylogeny.
Right.
And for a phylogeny, and I could see why this would cause a little bit, it'll be a little bit disconcerting. So this clarifies it a lot. Okay. This is really helpful. Thank you.
Oh, I don't have it handy. Yeah, so I can take this down now. But we will keep it handy in case we need it again. But yeah, so that's what we've covered so far. We've covered kind of the nested hierarchical scheme based on the pattern of common ancestry. So you want your domain, kingdom, phylum all the way down to reflect how recent your common ancestors are. And then we've looked at apomorphies, synapomorphies, right? Derived traits, and then plesiomorphies, ancestral traits, and how those terms are relevant to figuring out what that pattern is. And then we've talked a bit about all the examples we're using, right, to illustrate it are morphological, but then you do molecular and it's a little bit different. And I'm just going to look just for a second. I'm going to pull up some slides that I just used a couple weeks ago, talking about molecular phylogenetics, because we can talk about that if you want. I'm happy. And I figure for our audience, probably want to wrap this up. Sal, what do you think? Like 10:30? We'll figure like 90 minutes.
Yeah, that's fine. That's fine. Yeah.
People probably get bored after that point, but I'm happy to talk about this all night. But
no, this is good, because just like our last meeting, I had to chew on what you said, and then we met, and we're basically continuing the conversation we had on my channel.
So we've got that one.
So there,
we've got
this will be good.
Okay.
We can wrap this up. You've explained so much, and you also you helped me with like going back to reading rooting diagrams of rooting. So that might be the last thing. If you can say, okay, how do you know when you have a rooted tree when I'm reading?
Yeah. So here is a slide that looks at not just rooting versus unrooting, but also the how you can read a rooted tree and how differences in the tree affect versus don't affect it. So one of the things you can see here is, so when you have a rooted tree right here we go. We pull up this. Here we go. Okay. So when you have a rooted tree, what you're saying is you pick a group that's your out group, and you say this out group is representative of the ancestral traits. So like if you're comparing, for example, a bunch of different eukaryotes, you can use a prokaryote and say, okay, the prokaryote existed prior to eukaryotes, right? So the prokaryotic version of whatever trait we're looking at, we're going to call that the ancestral trait. And then in terms of the branching pattern and how things differ from that, from the ancient state, we're going to compare the differences from the prokaryotic version, and then we're going to see how all the eukaryotes sort out. So we're taking something that is less related to the group we care about and setting that as the baseline. And by doing that, we're setting an evolutionary direction. We're not just saying how many differences you have, we're actually saying which versions of a trait are ancestral and derived.
See, this is very helpful because I remember when I was asking Dr. Rogozin what, we were trying to do, I was trying to do a tree on the beta-lactamases, which were actually the nylon-eating beta-lactamases. And is really funny. He said, "Oh, that's a mid-rooted tree," and he said, you can root it." I said, I feel uncomfortable. And I said, "What's rooting?" And he said, "Oh, that's all." I remember in the lectures he said, "Yeah, rooting's a really tough. A lot of people will."
Right. So
I'm not going to go there.
So this slide. So this slide gets at the challenge of rooting when you're talking about molecular traits, because for every site in your genome, you have 4 options, right? A, T, C, G, like those are your options. So how do you know which one is ancestral? You can go back and forth, right? A can mutate into anything, C can mutate into anything. The rates are different, right? We know, for example, that C mutates into T a lot faster than any of the other changes happen. So like if you see a site that is a C in a lot of your clades, right? Or in a lot of your taxa, and then like you get a T in a few of them, you're like, that T, that's probably the result of mutation from the C. But like, you have to know stuff about the relative rates of mutations to be able to make that judgment, right? So with DNA characters, the rate of change matters, because if it's going too fast, right? If you're looking at, for example, hemoglobin, well, hemoglobin's a bad example because not all eukaryotes have hemoglobin. I'm trying to think, what's something all eukaryotes have, but it mutates really fast? Well, you can look at like a pseudogene or something. Pick some pseudogene, right, where it just accumulates mutations. It doesn't work. It doesn't do anything. And you could look at it and go like, well, there's an A for some of the sites. There's a G for some of the sites. Like, the A to G, G to A, it's approximately an equal rate. And like, it's hard to say really, and you're going to maybe need to use other data to resolve that question of what the ancestral and derived states are when you're dealing with molecular data, which is why you don't just look at one site or one gene. You look at, thousands of bases at a time, and you look at many, many regions. And this kind of goes back to what we talked about at the beginning, where you're not generating one tree, you're generating a whole bunch of trees and then building your consensus out of what you see.
I just realized, so like a C to T, now I'm embarrassed. So they have transversions and transition, and then one is a pyrimidine and the other is a purine, and I should know this, and I've forgotten.
That's okay. So A and G are the purines, so they're the 2-ring ones, and then C and T, and also U in RNA, uracil, those are the pyrimidines. Those are the smaller 1-ring bases. Okay. A transition is when it's a mutation from a pyrimidine to another pyrimidine, or purine to another purine. Okay. And a transversion is purine to pyrimidine, or the other way around. So like A and G.
Transversions are harder.
Transversions are harder, yeah,
okay.
Because transitions can occur through simple like chemical changes. So like C to T, all that is a deamination. and your C turns into a uracil. That's all that is. But to get a C to turn into an A, you actually need to replace the pyrimidine base with a purine. So that's less likely. So the diagonal arrows here are transitions. The vertical and horizontal arrows are transversions. The transitions are more rapid than the transversions.
Thank you. This is starting to crystallize stuff I was just kind of wrestling with. I know that was peripheral to the point, but you were saying
no, that's good. Because
the decisions for deciding what should be your outgroup can affect the clades, basically the grouping of your clades in
right.
your tree. Okay.
Right, because if you make a judgment call and say, okay, I have a T at this site, right, and we're going to say the T is the outgroup. We're going to say that's representative of the ancestral state. And some of your taxa that you care about have a C at that site. You're going to get one set of relationships, right? Because that implies a T to C mutation happened at some point, right? If you go the other way and you say C is the ancestral state and a C to T mutation happened, you're going to get different trees because C to T happens a lot faster than the other way, T back to C, right? So that's going to have implications for what are the more likely patterns of a divergence in your tree.
Yeah, and I could see why I wanted to avoid this, because proteins are so much easier. I don't have to deal with the introns, and
yeah, right.
But then you're missing all this richness here. That's probably why I didn't want to deal with. Now I can see why I went into the protein world. I didn't want to deal with the genetics.
Oh, here we go. Here we go. So this is a useful. So this is useful. Another thing, just sticking with rooting and kind of what rooting does. When you root a tree, what you do is you put an invisible time axis essentially on your tree, right? So if you look at this tree, this is just random tree. It's totally arbitrary. You've got taxa A through G, and this is a rooted tree. So like each node represents a common ancestor of the things descended from that node. So like this node, right? Help if I use my cursor correctly. This node right here represents the common ancestor of C and D. This node right here is the common ancestor of E, F, and G, so on down the tree, right? Back here, this is our root. This is the common ancestor of everything on the tree. So by rooting the tree, we're essentially saying from bottom to top, we're going from the past to the present. If we use an unrooted tree, so if we represent the tree like this, we're not making a judgment call over what represents the past versus the present. We're just saying differences between them. And you can root that tree at any point. You could root it here, you could root it here, you could root it over here, right? You're not making that judgment call. Once you root it and you say this represents the most ancestral state, now you've set your out group. And actually, I know I'm being all over the place with the slides, but I want to go back to, so I'm going to put this down. I want to go back to the slideshow I showed a few minutes ago, because it has what I think is a really cool way of showing the implications of different rooting patterns. So like why you have to be careful when you root and the importance of that decision in terms of making that judgment call. So let me put this up because we looked at this before. We can root this in all different places. So let's say we root it here. We say A is representative of our ancestral state. So we say the common ancestor of everything on this tree is somewhere between A and node 1, right? So now we make that the root. A is our outgroup. A branches off before anything else. We come down to node 1. It's right here. You've got B going one way, and then the CD branch going the other way with that black line in between. And then you go down to node 2, and you've got C and D. Does everyone see the pattern there of how you can translate from unrooted in the middle to rooting at A right here? Setting that as the root, and then everything descended from that based on this pattern. Is everybody okay with that? Sal, are you okay with that?
I'll have to digest it, but go on. I think I'm seeing it now. Yeah.
So you can do this all over the tree. You could do it with B, right? And you get a different pattern, right? So now you get B as the root. A branches off first, then C and D. You could do it for C. You could do it for D. It's each time you get a slightly different pattern of the tree. Or one more way of looking at this, you could do midpoint rooting. You could say the root is somewhere in between these two groups. So you get a clade that's AB, right, as sister taxa, and you get a separate group that's CD as sister taxa.
This is actually, I got to tell you something. This is very enlightening. Like, I didn't realize. Okay, so let's go to that diagram where A is the root, and then it has that line kind of going up where the
so here,
dark
we'll just show that. Yeah.
That doesn't necessarily mean it's mid-rooted then.
It's not. So this is explicitly not midpoint rooted. Midpoint rooting is when you take kind of the consensus sequence of everything and say whatever is the kind of average of all the taxa you're dealing with.
All right.
Take that consensus, that becomes your midpoint root. That's different from what we're showing here because go ahead.
So what is the unrooted tree? Is it that one, that star diagram?
This is the unrooted tree right here.
If you could point to, I can't see it on the.
Oh, I'm sorry. I had my mouse on the StreamYard stream, not the.
I know. I do the same.
There you go.
So that is.
This is the unrooted tree right here.
Okay, so this is actually kind of important because I've heard a lot of arguments over Nathaniel Jeanson's work and John Sanford. Okay.
Yeah.
That starburst diagram then is the unrooted tree.
That would be this starburst diagram.
Right. That's an unrooted
tree. There's nothing here that sticks out where we say this is the common ancestor that everything else is descended from.
So we would call that an unrooted tree.
This is an unrooted tree. Yeah.
I've heard. Now I could generate these in Mega for anything I can put in there, and so that's what we call. And so when you have that one with the like the bars, that is a rooted tree.
Right.
Okay.
So you can, like here, right. So this is what Jeanson shows. And
that's an
Dr.
unrooted
Rob Carter has a very similar tree in a piece that he wrote. It's very similar to the tree that I just showed. That's Jeanson's tree. It's an unrooted tree because there's no branch on that tree where they're saying this branch is the common ancestor that everything else is descended from. They don't make that judgment for anywhere on that tree. So it's an unrooted tree.
Okay.
Right.
All right. So now this is clarifying a confusion I've had for like the last 4 years. So this is very helpful.
I'm happy to. I live for those moments. Like that is when I get when I see the light bulb go on. That's
this is why I said like I said, I went into the protein field because I I didn't want to deal with this.
Let me see.
Okay, this is very helpful.
Here we go. So I actually have I have a PDF. I've downloaded EvoGrad's replacing Darwin, all his blog posts on this, and this is the one where he very nicely actually shows the rooted and unrooted tree. So here is the difference, right? So what we have right here, this is the same tree I just showed, right? And you've got your 3 nodes that Jeanson identifies as your 3 the wives for Noah's sons, right? He says these 3 nodes represent the 3 wives, right? 3 clades descended from those. Like that's how you can fit this into that story. The problem is the lines here that connect these nodes, anytime you see a line on an unrooted phylogeny, or on a rooted phylogeny for that matter, anytime a line connects 2 branch points, that means that one of those branch points, one of those nodes, represents a population that is descended from the other one that is connected to it. So what we have here are not 3 coexisting common ancestors for 3 separate clades. What we have here are a series of ancestor-descendant relationships. So like you can root it at this one up top, the pink one, but then the other 2 are going to be nested within that, right? It's going to be a pink clade, and then the black one, and then the green and blue one. You can nest it in the middle, and then you get the black one, right? But then separate from that, you have the pink one and the green and blue one, or you can nest it at the bottom, and it's the reverse pattern.
Could we also mid-root it and then?
So if you mid-root it, you're going to end up with something around here, right? Something like here. I actually, let me show you a simpler version of this because I actually made this. So here is my so here's the version from one of Jeanson's posts on AIG. It's this one. And so what I did here is I took A, B, and C. We're going to take those 3 nodes. We're going to label them A, B, and C. And depending on where you root it, you get different trees. So here is the simplified version. We cut out like a lot of the branches, but just to represent it, this is Jeanson's tree. with those same three, A, B, and C. So we can root this in all different places. This is what the Jeanson's claim is, right? You get basically 3 nodes, and then you have kind of 3 separate clades descended from those. You can root it at A, and you get this pattern, right? You could say A is the origin point, and that works for anything beyond A as well, right? You could root it like way down like this branch, right? And you're still going to get this pattern where A branches off, and then B and C are nested within that.
So I could take any individual from A's in the A group, and that could serve as an out group, and we basically get this. Okay, I learned
right, and you would get something where you would put A then if you were to take like this branch right here. So I'm rooting this right here. We're saying this node is the root, right? Is the judgment call I'm showing right here. But you could also say like this branch right here is the node, and then that just puts A would be like down here, and then B and C would be kind of the way they are.
So just to conceptualize this,
or A would be
if I were like saying, again, I'm sorry to harp on my
don't apologize. Don't apologize.
It happens to be the one that I'm most familiar with. Hypothetically, I could take any bacteria to be the out group for the topoisomerase. We would probably would not affect the tree structure.
It would probably get pretty similar, yeah. Right.
Okay.
So then, so going back to this, you could root at B. This is the pattern you get. You could root at C. You get the opposite of the A root. You could also root in between, and this is where we get to midpoint rooting. So you could go in between. So instead of picking one of those nodes, you go in between those nodes somewhere. So you could do it between A and B, and this is the tree you get, where C is descended from B. Or you could do it the other way, where A is descended from B. There's no way to take...
So mid-rooting is still considered rooting, is that correct?
Yeah, because again, you're not picking a specific node and saying like that's my root. You're saying in between 2 nodes is where the root goes.
This is very, very helpful. So now if I could just ask a really dumb question, and I wish I had my I'm gonna have to try to describe this with words. So we see with the A to the B, you have that bar connecting the A to the B in the lower part.
Down here.
You don't necessarily have to have that extra line there that's pointing up.
This one, so that's the root. So what you're saying is right here is where the common ancestor is. So we're going to say, boop, we're going to just draw a little line off that and say, that's your common ancestor. And then we're going to show the rest of the tree descended from that common ancestor.
Now, if I could add, could I share my screen just briefly?
Yeah,
this is
100%.
where I'm confused. This is where I'm a little confused. So,
here you go. Let me pull this up right here. There you go.
How come I don't have that line pointing? That's
it's
what
arbitrary.
I don't.
You don't need to show the little thing. It's the structure of this tree is such that, remember I showed you how when you root a tree, you imply the time axis. You never show it, but you imply it. What you have is from left to right here, you have the time axis from most ancestral to most derived.
So that little line is kind of like,
yeah.
Okay, I did not know that.
Yeah, it's most people will include it in a rooted tree as a way of just kind of saying like, here it is. But so for this tree right here, so the 0.2, that's genetic distance. So that's not time. That's actually genetic distance. And that's why the branches are all different lengths. And in between nodes, you have branches of different lengths. It's showing you the different, basically the number of genetic differences between those different groups. So for that one, you don't put that root on it unless you're saying from the common ancestor of everybody on this tree to the first branch point with your out group and then everybody else, how many genetic differences are in there, right? So for this one, you wouldn't put that there because you're not measuring time, you're measuring genetic distance. If you're measuring time, if you can say like approximately when that thing happened or relatively how long that time was compared to the other divergence events on the tree, then you can put it on. Or like the ones I showed you that I use in class, if you're just doing it kind of, schematically and it's not a technical thing in terms of either time or genetic distance, then you put it on because it makes it easy to say like, boop, that's the starting point. But you don't need to have that to have a root.
That is just immensely helpful because, this is one of those things in the class. It's like, we're focusing on so many things and it's just like, okay, did I misunderstand that? And you just kind of carry it with you because this this is really more bioinformatics. It was not phylogeny. And I'm like, okay, this is, we were being taught,
And like we were saying off air before we started, if you don't like study this thing specifically, there's a whole bunch of language that just like it's gibberish because you just don't use the language day to day. So for someone like you who's like, let's do protein stuff, right? And you're gonna turn around and go, okay, let's do this with phylogenetics. Like now you have this whole other set of vocabulary. Now, like I'm more familiar with the phylogenetic stuff, but if I turn around and try to do the protein stuff, I'm like, I don't know. This is not my field. I don't know what I'm talking about. So you have to kind of like figure it out, and that's what we're doing. And it's fun.
This is immensely helpful. I mean, because I now I feel like I can start to read evolutionary literature a little bit more clearly, because actually when I was hearing the usual stuff going on the internet about rooting and unrooting, I'm just like, okay, I feel kind of bad because I should know this and I don't, because that's just not my specialty. And so all the arguments about the criticisms of Nathaniel's work and then all the back and forth, I'm just like, I had no clue with the term. I said I didn't, for one thing, I'm not going to weigh into it. I mean, Nathaniel's my friend, but
like so you should put in a good word and say we should talk. The three of us can talk. You should put in a good word. For me,
we can. Well,
I'm only half joking.
Just for the
I'm actually serious. That would be really cool. I would love to talk to Dr. Jeanson. I think that would be really cool because I got so many questions.
All right. You know what? I will put in when I see him.
I appreciate
we occasionally, the last time I saw him was on a conference, and sometimes I will refer evolutionary biologists, and so I'll say this would be or at the very least I can have a conversation with him.
Bean was wanted attention.
Yeah.
So
I wanted to say I wanted to thank like Ethan. Is Ethan Baldwin an evolutionary biologist? I wanted to thank him for some of his side comments.
I don't know, but yes. Hi, Ethan.
I've
been following the chat as well. I also see a bunch of questions in the chat. I see Andrew. I don't think we're going to have time because we've been going for almost 90 minutes now. So I think around another like couple minutes we'll wrap this up.
We could probably talk about like what resources, because I'm not.
Yeah.
Honestly, I could go and take classes now, but I have so much of a backlog just to relearn everything that I'm supposed to know that I've since forgotten that I'm probably not going to have a chance to take a evolutionary biology class anytime. So, but if there is a book, if you ever post, if I could just suggest this, if you ever post your lecture notes or something, I would be delighted to read through them if they're public domain. For your biology.
So I have to keep doing I have that Evolution for Creationist series that are basically my lectures but cut down, and I have to keep going with that series. I kind of when I went back to work after child number 2, it got really busy, and so I kind of put that series aside. But I do need to get back to that because that's a really fun series, and I'm building up. I'm almost through the genetic stuff, and then I'm building up to the phylogenetics and like the mechanisms of evolution. Like I promise everybody, I'm getting there. I plan to do it. It's just that one takes a lot more work than some of the other stuff I do because it's like a half-hour lecture that I have to prep, and it's not like a 10-minute video, so it just takes longer to set up. So I will do more of those.
I'd be delighted to. I'd be delighted to see it. And you have unscrewed kind of my misunderstanding for over the last 4 years, because it's been 4 years since I took Dr. Rogosin's class.
Oh,
so thank you.
I'm here. Okay, buddy, you go. Good boy. Just let him out of the room. The dog runs the show. Okay.
This is, if you want to just address the things in the side chat, I'm content. This is great.
So I think this is a really good place to stop it. We've been going for like 80-ish minutes. So I think that was really fun, informative. I just love talking about this stuff, but I don't want to keep everybody all night. I can't do the long streams like you can just go. I don't have the stamina to go for that long. So I think this is a really good place to wrap it up.
I don't have a teaching schedule like you, so that's probably why I'm
true. So I will just say, so first, let me just thank Sal for coming on. This was a really fun conversation. And I want to say I really appreciate Sal as a young Earth creationist who is like, Sal, you're really trying to like grasp the stuff that you don't have formal education in. And I appreciate that. So thank you. I really like these conversations. I think this is wonderful. And I would love to do more of this. Happy to come over to your house. Happy to have you back at mine, however you want to do it. I would love to do this more in the future. Second thing is, Sal, do you have any fun stuff coming up on your channel?
Not related to the creation evolution controversy. I don't have anything scheduled. So actually, you're the next fun thing, because when I rebroadcast, that'll be the next fun thing. Go. So short answer is, we have some archaeology stuff tomorrow, God willing. And then Saturday's a variety show. And if people have seen my variety shows, it's all over the map. It usually is. When I clean, as I'm reorganizing the house, whatever book comes up, I said, that's going to be on the variety show.
Nice.
So
the whole stack.
Yeah. So there's nothing pre-programmed at this point. I'm avoiding the debate stage because it just, this is far more fulfilling.
I agree. I really like these types of conversations. These are much more valuable than the adversarial debate. I know you said via email you didn't watch the debate with Kent. I can't blame you. That was purely for fun. That was like everyone knew that was going to accomplish nothing. It was 100% just for
and maybe
that was not a productive discussion, right?
Yeah.
So coming up on my channel, actually, I'm so pumped for this. I am having Dr. Michael Behe back on. We're doing another round. So we previously, way back in March, we talked about Darwin's black box and irreducible complexity. And just in about a week and a half, it's July 7th, at 1 p.m. Eastern, we are going to talk about his second book, Edge of Evolution, which has to, rates and how, different examples of adaptive evolution and the rate at which things can happen. And if there's a limit on that rate, I'm really excited for that conversation. I had a lot of fun talking to him back in March. So we're going to have another one. And that'll be on July 7th at 1 p.m. right here. And that link's available. If you go to my channel, check it out. That'll be Dr. Michael Behe back on.
I'll promote it. I'll promote your show.
Thank you. Yeah, please.
Down, July 7th.
Yeah, July 7th. Yeah. We're doing it. I'm excited. Dr. Behe he was real good sport the first time around. We're gonna have another fun conversation.
You hammered him pretty hard, and I'm like, it's funny. I was just like, Dr. Dan, you kind of showed why I don't like using irreducible complexity in my arguments. I took a little bit of flack for that, but I thought that was fair. That was a great show. I'm glad he's coming back. That means he likes you.
Right. So because again, it wasn't a debate. It was a back and forth, and we agreed on some stuff, we disagreed on some stuff, and it was informative, and it was more productive than if we went on MDD and just had a back and forth debate. So that was I really like this format. I'm trying to get more people into this kind of format than the debate format, because the debate format it's maybe entertaining, but it's terrible for conveying ideas to people. Whereas this format is really good for that. So that's that's what I have coming up.
I could say here, I don't know how that. Can you tell me how? Can you explain that to me? In a debate, you don't do that.
You can't say that. Right.
Sad. Yeah.
Right. You can't do
so
that.
That's kind of like, this is why I like this. This is where we can actually move forward. And, I get flack for this, but I just wanted to say to the creationists out there, see, I hope you learned something, because when I read broadcasts, I want to say this is why we do this. Too.
I hope you learned something, because there's stuff to learn and it's cool. And phylogenetics works no matter how old the world, it still works. It's still, so understanding how it works is important. All right. So I think with that, we're gonna call it a night. So thank you everybody for watching. There's still 21 people watching at this point. Thank you all for sitting here while we talk about phylogenetics for 80 minutes on a weeknight. That's awesome. All right. Thank you again, Sal. Thank you everybody for watching. Have a great night, everybody. I'll see you all soon.